Right, this video looks at an OCR past paper. It's 2017 AS level, breadth in biology, paper 1. I'm not going to go through the multiple choice in this video, might do in a later video. And to keep the video down, the length of the video down, I'm not going to spend too long explaining things. I just want to go through the answers. So first question 21. A student looked at a slide containing onion root tip cells under a light microscope in order to identify cells in different stages of mitosis, which they show there. And then they got some kind of measuring bar down here at the bottom. Explain why onion root tips were used to view cells undergoing mitosis. Well, the main reason is that they're growing and therefore undergoing mitosis. So if you think about a root tip, they spread out to seek out water and minerals and the like. The other thing is that because they're underground, the obvious thing there is, well, it might not be obvious, but if I say it has no chloroplasts, these would get in the way of an otherwise clear field of view. A lot of plant cells, obviously above ground in the leaves, many chloroplasts that would obscure such a view that you can see there. Drawn rather nicely, I might say. Stain, you'd use to stain the nuclei. This one, methylene blue. There are a couple of others, I think. The student used an eyepiece graticule, which is shown in the figure above there. There's that scale there. And he calibrated it before carrying out a root tip squash. He observed that we got 20 micrometers equals 2.35 divisions. Somewhat confusing. I think what that means is that that's division. I think that scale will be about right, be 20 micrometers. So 2 large divisions and then a 3-5, because you wouldn't get that level of detail if we're talking about, the tiny divisions. That's the kind of sense of it. I measured this before I was going through this paper with a student. I remember this is 14 millimeters. Can't do it on here. The scale's changed on this virtual copy here. So I think 20 micrometers, if we say that's 23.5, because, if you're measuring 14 little divisions, and then there are 23.5 little divisions on the bottom, then you now made it into a fraction. That's a more complicated way of saying that this is a bit smaller than this. That's the way to think about it. And what does that give you? That's a calculator. I've left it in stats mode. Well done. NU1. This is the degree by which it's smaller. Sorry. But you've got to times that by your 20 micrometers to get it proportionately smaller. I got 11.9. As I say, I can't really, stop and explain everything. Just maybe that gives you a sense of how to look at that maths question. You can ask if you want fuller details. 22. Many multicellular organisms need to be able to convert monosaccharides into polysaccharides and back again. They do. We do it all the time. Mammals convert the monosaccharide glucose, a sip of coffee there, into a highly branched polysaccharide called glycogen, which gets stored in liver cells. Explain why mammals store glycogen instead of glucose. Man, they always ask this. Right, it's insoluble. You should just know this, really. You've got to be able to trot this out. And therefore doesn't affect water potential. Otherwise the cell swells up and bursts, so you don't want that happening. You could say it's highly branched. So this is explain. So you can't just describe. Highly branched, and so accessible for breakdown. Sorry about my writing, hurrying. If you imagine like glycogens, the strings of alpha glucose units stuck together, but branched off with the 1,6 glycosidic linkages as well. And so when the enzyme comes along, it can release a lot relatively quickly. And because of that structure, it's also compact, so it doesn't take up a lot of space in that form. There's probably some other bits in there, but I'll just keep it quick. Three mark points. You don't need to write all this. You just need to keep it. What you're trying to do is maximize points per minute. So you want economy of language. Learn to speed this up. And if it means drawing a diagram, draw a diagram. I used to mark these exams. If I see a diagram, I can still mark it. So B, but do write it out afterwards in the exam. Okay, you've got to communicate with the examinator. Cross between terminator and examiner. You've got to communicate with the examiner. If they can understand what you're saying, they can give you marks. B, humans use the enzyme alpha amylase they don't make the decision to use it, it's badly written, to break down polysaccharides in food for absorption into the blood. The gene for human alpha amylase is found on chromosome 1. The gene is transcribed in the nucleus, and translation occurs on the rough endoplasmic reticulum in the cells of the salivary gland. Describe how the molecule is prepared and secreted by cells of the salivary gland after translation has taken place. Okay, what they're asking about the Golgi apparatus definitely here. It's part of the answer to this. So in the Golgi apparatus, you get modification of the protein. After it's been transcribed and translated, the gene, that is. So it could be further folding. Could be like adding carbs, even lipids or whatever. That's the first thing. Then it's packaged after that. After it's been processed, it's packaged. And then you'd say into a vesicle. This being, a little membrane-bound sphere. And then it'll be transported and then say how it's transported. It's on the cytoskeleton. Just keep it brief. And then to plasma membrane. Don't write membrane, write plasma membrane. Hair on the screen, damn it. Plasma membrane. And then it's released through the process of exocytosis. That means outside the cell. 23. The fluid mosaic model describes plasma membranes in all living organisms. 23A. How does the fluid mosaic model describe the structure of plasma membranes? Okay, this is basic stuff. You've got a phospho, so you need to put these words down. Phospholipid bilayer. I guess you'd say, but keep it brief. Okay, hydrophobic fatty acids inside. Hydrophilic phosphate head group. Don't worry, if that doesn't describe that part of the lipid precisely, it tells the person you're communicating with that, this is the active component that gives it polarity in aqueous environment. Fluid, this phase is fluid. It just means that the phospholipids shuffle about. Mosaic, so proteins are embedded within. How does it describe? I guess they're sort of interspersed. Something like that. It's only 2 marks. Plasma membranes are partially permeable, allowing some molecules to cross the membrane. This is actually, all this stuff is really standard for the AS. It's just applying it, really. With relative ease, one molecule that crosses membranes is the steroid hormone, progesterone, which is produced in the ovaries from cholesterol. This is a lipid. So this is about being lipid soluble. Explain why progesterone can move across membranes. It's lipid soluble, therefore not repelled by hydrophobic. Must improve my writing. I'm just trying to get through it in a reasonable amount of time. That's those 2 marks. I did have a quick scan of the mark scheme. Name one other molecule that can cross plasma membranes. Badly written again. I think what they mean here, and I know from the mark scheme, is that it's not going to go through a protein channel, although that question doesn't explicitly state that. This should say lipid bilayer. And that'd be a very small molecule like oxygen, water even, it's very small. Some can get through. Let me just double check the mark scheme on that. Let's hang on a second. That's a silly question. Water. Carbon dioxide they get. What do they say here? Alcohol does. Vitamins. Yeah, anything charged, it's not going to get through. Anabolic still. They allow a bit of imagination on this one. Okay, I was just curious. Sorry. Let's get on with it. Where were we? Potassium ions, so part C now. Potassium ions are unable to move across membranes as they are charged. State how the structure of the cell membrane allows potassium ions to enter, leave a cell. Be a protein channel. Be a channel protein or a transporter protein. It's got to go through protein. C2, the process of active transport uses ATP to pump potassium ions through the cell surface membrane against the concentration gradient. ATP is made up of phosphate groups and two other molecules. So it's when they kind of mix up the question a bit, they do this. ATP. Do a quick sketch, maybe. Looks like this. I'll just put it simple. It's got these three phosphate groups. I said I wasn't going to explain it. But you should know that you got this. So this is very much like a nucleotide. And so you got the base similar. It's the same as you get in DNA and RNA. So it's adenine. And this is ribose. So, yeah, it's the same as you would get in RNA, but not DNA, this bit here. All right, going off on a tangent. Sorry about that. Okay, so this is where, like, maybe non-math students might panic a bit, think, oh, Christ. Right. D, a group of students investigated the effect of temperature on the membranes of beetroot cells. So this is an experiment you might have done if it had not been for the pandemic and you were kept away from school. But you might have done it, I don't know. A colorimeter was used to measure the concentration of purple betalain pigment. Let's just call it pigment to simplify. That leaked, can't talk, out of the cell when they were exposed to different temperatures. Okay, so they measure the absorbance, that's standard deviation. They measure the absorbance, it's increasing. But also, the variability of this data at the top is also, because it's a wider spread. Using the student's t-test formula below, calculate the value of t for the data between 50 and 60. Right, okay, this is just quite a lot of legwork to get these 3 marks. Okay, so we've got a mean here for A, which is 0.21. And then it says minus the mean for B. It's B here. A before B, except after. No, just always before B. Minus 0.44. They put it in brackets. And then we got a bunch of stuff here. So S is the standard deviation. SA is that one. So you've got 0.06 squared. N then is the number of readings, which is 10. Divide that by 10. Plus the second one, which is 0.44. Squaring and then taking the square root. This all gets rid of negative signs, I think. Don't quote me on that. I don't teach much maths. Right, 0.44 squared. It's over 10 as well. So this top one is see here, like minus number 0.23. Then within the square root, you got, let's put brackets in, hang on a sec. Got 0.06 squared divide by 10 brackets plus brackets 0.44 squared. Possibly I should have done this before. 10. This comes to this value. Come on. But you've got to square root that. So that square root is answer. So 0.23 divided by answer. It's possible I made a mistake. Okay, so I need to revise my stats. Problem here is this is giving you a negative value. Okay, I made a mistake there. That must be A, which goes there, and this must be B. Hey ho, we all make mistakes, even me. I did it the other way around last time. Because this was a tricky question for me. I don't, maths. I kind of know what I need to apply, but I got this wrong. Nobody's perfect. Okay, so in this case, that's B, that's A. I need to go and have a look at that again. However, that's the right answer. The critical value for T at the significance level of 5% with 18 degrees of freedom is 2.1. use the value of t that you calculated to explain whether the null hypothesis should be accepted or rejected. This is always the same. So you say value or calculated value above critical value, therefore reject null hypothesis. So data is significant. You see, I know all this, but to use this, God damn them examiner, sorry. There's no point complaining. I should have looked at that properly. Right, still annoyed with that. E. Students plotted the data onto a graph shown in the figure. Okay, so we've got describe and explain the pattern shown. So you could say that increase in temp to 30 degrees C, little change in absorbance. And I guess you say from 30 up to 60, increasing rate of release of betalain, or just say pigment, which increases absorbance, something like that. You've got to describe this is fairly flat, this goes up, and this is because the membrane is breaking down at these high temperatures, releasing the pigment, which causes the increase in absorbance up here. Don't get too bogged down in figures, just keep it brief. 24. About classification. In 1990, Carl Woese, I'm not sure how you pronounce that, suggested a new top-level taxon, as domains, to the current system of classification of living organisms, which he termed a domain. It was based on DNA and RNA. He used the result. Okay, all right. It's based on RNA. He used his results, which comes from DNA, from studying RNA to organize organisms into 3 distinct groups. I don't like this question. Name the cell component that appears in organisms of all 3 domains that Woese suggested. I guess it kind of makes sense. Well, that's a ribosome. Because the ribosome is the one organelle that's associated with RNA. One of the domains he suggested was eukarya. So the other 2 are bacteria and how do you spell this? Archaea? Never was good at spelling. State 2 defining features of all members of the domain eukarya. That's the eukaryotes. They have a nucleus, a membrane-bound organelles. There's other things as well, like ribosomes, different, whatever. Woese carried out a detailed study of RNA molecules in order to draw his conclusions, suggests two ways in which the scientific community are likely to have validated Woe's research. Well, go and repeat it, check it out, just don't believe him. Repeat experiments. And then you could develop further experiments that would kind of elucidate things a bit more. And then all get together, have a great time at a conference and discuss it all. Right. Sorry, it's going on a bit, but I will get through it. You may wish to pause the video and make a cup of tea. $25. Spat my coffee on the screen. 26. And then the end. 25. Semi-conservative replication describes the process by which DNA is replicated in all living organisms. Explain the meaning of the word semi-conservative. So one strand of DNA. God, that's shocking. DNA. So it'd be like the coding strand or non-coding strand. That's just to explain that what I mean by strand. You know, with all the nucleotides attached together in a long string. Conserved. And bonded to new nucleotides. That's what, so, you know, if you need to go back and look at semi-conservative replication, then you just need to go back and look at it. DNA ligase. All right, this is used in genetic engineering. If you imagine you're going to stick this into a plasmid or something, you want to join this on the end, like a blunt end, like this. Ligates tied together. Do not get this confused with the other enzymes involved here. So it says state two other enzymes involved and describe their functions. Helicase and DNA polymerase. This one then unzips. So it's breaking those hydrogen bonds between the bases. You know what I mean? Just got very simple. And then state two and describe their functions. So it breaks hydrogen bonds. Allowing new nucleotides to assemble. I guess that's the word. And DNA, so I think, in the mark scheme, that's a mark, that's a mark. What does it do? That's a mark. This then rejoins the sugar phosphate backbone, so it enables formation of Let's just move this up a bit. It's getting hard to write down now. Diester bonds. It's that one. Okay. Michelson and Stahl. In 1958, Matthew Michelson and Franklin Stahl carried out an experiment that provided evidence to support the hypothesis of semi-conservative replication of DNA. Michelson and Stahl grew E. coli bacteria in a growth medium that contained only the heavy isotope of nitrogen. Right, nitrogen 15. It's heavier. It's got an extra neutron. It's heavier than a normal nitrogen, which is nitrogen 14. They transferred the bacteria to a growth medium that had the light isotope. I thought this is the normal one, actually. And this is the heavier one. I don't know which predominates. I think it's this one. It's not a chemistry lesson. And allowed bacteria to undergo cell division. After each division, the DNA from some of the bacteria was extracted from the culture and centrifuged to separate it. And in figure 2.25 shows the bands of DNA in the centrifuge tubes after a specific number of divisions. Draw it over here to kind of explain things a bit. Okay, the tube labeled generation 0 shows a single band of DNA containing bases that contain only heavy isotopes. And you can see it's gone further down the density gradient here. It's a denser element. This one in the middle, it's higher up. Explain how the results from the other generations provide evidence to support the hypothesis that DNA replication is semi-conservative. It's quite difficult to explain that in words, but with a diagram it kind of helps things, which I'm trying to do now. Higher up, less dense, therefore has this isotope. Now we know it's 50-50, that isotope. How do we know that? Because higher up again, higher still, and so you've got a band, you've got two bands. That band is pure nitrogen 14, which is why it's higher than this one, which is a hybrid between this and this. Draw a diagram and then put it into words. Sorry, I'm just trying to keep it short, that's all. This is the last question, thank God. Right, okay. Right, 26. The protease enzyme bromelain can be extracted from pineapples. Well, that's nice to know. A student investigated the effect of changing the concentration of the enzyme and measured the time taken to break down the protein gelatin. Okay, so this is a protease. State 3 variables that the student would need to control in order to make the results of the investigation valid. Concentration of enzyme, independent variable, and then what they're measuring, time dependent. So these are all control variables, so like the one to trot out every time is temperature. Just state them. Okay. Well, you could have volume. You can't have concentration of the enzyme. That's the independent variable. But you could have the volume that it's in. You could have concentration of gelatin. It might be in solid state though. Still applies though. If you're going to say temperature, say pH, this kind of thing. The data from the easy mark says the data from the student's experiment is shown in table 26. So we got the concentration, the bromelain, and then you got the rate of protein digestion, and then you got standard deviation. Stats again. Look at the difference here though. This is a big error bar. This is small error bar. Showing, I say error, I mean standard deviation. Describe how the rate of the reaction was calculated. Well, a rate is per unit time. So this is God damn it. Rub this off. This would be conch gelatin. How that concentration decreases over time. Explain what the standard deviation. Oh, that's that question. So explain what the standard deviation shows. It shows spread of data about a mean value. So, to get good data, you've got to repeat the experiment several times to calculate a mean. Then you can calculate standard deviation and put error bars on the graph to show the variance of the data. This, as I was talking about here, shows that you get a lot more variability, or it's more variable in data at higher concentrations, I would say these three. I'll just say at higher concentration. And then, which you can see nicely on this graph, these are the error bars. There's tiny little error bars on there. And then these are big, comparatively big. Figure 26 shows the results plotted on a graph with the standard deviations as error bars. So, okay, the words kind of interchange. I don't, stats, whatever. Okay, so up the y-axis, rate of protein digestion per second. Along the x, concentration of bromelain. Explain the pattern showing the data using table 26 and the figure. Well, just draw on there. So you can say, as conch enzyme or bromelain or whatever increases, so rate of protein digestion increases, but it plateaus. How do you spell that? Plateau, it's a French word. Plateaus. Say, but starts to plateau from progressively flattening, whatever, however you want to say that. Explain the pattern shown in the data. Explain. So explain means what's actually happening with the enzyme at higher conc, more enzyme substrate complexes, greater, to a point, greater rate of breakdown of gelatin. Breakdown. Okay, sorry about the error on the stats there. I should have looked at that before putting this video up, but what the heck, even me, well, especially me, I need to go back and look at things occasionally. It's just the way it is. Don't stress about it. Just, I would leave you with this thing, really, that is doing well in exams is obviously knowing what you're talking about, which is a great deal of work to get to that stage. And then you practice these papers. But what you're looking for, you want to answer these questions obviously accurately. You want to use the correct technical terms as best you can, spell correctly. But you don't want to waffle on about stuff that's not relevant. You've got to get it right down to the bare necessities. And then hopefully you'll communicate what you know. And if you can draw a diagram in the exam. It helps. Okay, hopefully that's been of some use. Any questions, just let me know in the comments. Thank you.